variant proteolytic disease. Good, you guys. Avian influenza carried by a bird, and we come in contact with that bird, and we get infected with avian influenza. Is that bird a reservoir? Yes, it is. Okay, so those two examples, the mad cow prions that can cause variant proteolytic disease, avian influenza transferred from bird to humans. Those infectious diseases of humans that have non-human animals as a reservoir, they have a fancy name. They're called what? Zoonoses. Okay, so put a star by that, you guys. That's a vocabulary term. So zoonoses are infectious diseases. It can be transferred from non-human animals to humans. Okay. All right, very good. And then the third reservoir that's recognized is the environment. Now, for a pathogen to use the environment as a reservoir, these have to be very tough, resistant pathogens, right? A great example would be the endospores of some bacterial pathogens. So anthrax endospores. The endospores can survive in the dirt for years, maybe centuries, right? Endospores of Clostridium tetani that causes tetanus. Endospores of Clostridium botulinum that causes botulism. Cysts of protozoa that we study in labs. Fungal spores, right? So these are all examples of pathogens that can easily survive outside the host in the environment. Okay? And certainly if there's an outbreak of infectious disease, we're praying that the pathogen does not have the environment as a reservoir, because it's very difficult to control. Okay, so that's the first trait we need to have as a reservoir. Now, the second characteristic of pathogens is they must be able to leave the reservoirs and reach a new susceptible host. And that process is referred to as transmission. So that's step 2 there. And again, you guys, we're going to hit this so lightly, but we do want to get some key terms in here. So with transmission, some of the key terms I'd like you guys to be familiar with is, first of all, we classify transmission with regard to the direction in which the pathogen is transferred. And probably the most devastating type of transmission is what we call vertical transmission. And if you think of vertical, you think of it as, I think of it as like this up and down direction. So vertical transmission by definition is transmission of pathogens from one generation to the next. And we'll just make it more specific. Vertical transmission, we're going to say, is when a pathogen is transferred from an infected mom to what? To her baby, right? Okay. Now, this can occur depending on the pathogen. Some pathogens can cross the placenta, infect the baby in utero. Often that's devastating because the baby's so sensitive to damage when it's developing in utero. It can occur as the baby is being delivered through the vaginal canal, as we said, with genital herpes. Babies at high risk for genital herpes. Moms that have genital HPV, the baby can get infected with HPV when it's being delivered through the vaginal canal. And we also know that babies can be infected through breast milk. The story of mom who's HIV positive, delivers a baby. The baby is not HIV positive. They've shown that baby can become infected with HIV through ingestion of HIV positive breast milk. Okay? And just another herpes story. Those of us that have the oral herpes simplex viruses and we're kissing, that newborn baby, we can infect a newborn baby after birth too. Okay, so these are usually, as we said, often devastating infections because the baby's immune system is still very immature. It's not very strong yet. Okay, so those are we want to really try to avoid those. And then just to simplify our life, you guys, I'm going to tell you anything that's not vertical is, what's the other direction? Horizontal, right? So the reason that, this one generation to the next is misleading is you guys know that I'm definitely of an older generation than you all. Okay. But if I had active TB right now and I'm coughing mycobacterium tuberculosis into your faces and you're inhaling them and you get infected, even though I'm a much older generation, that still would be considered more horizontal transmission. Okay, so just think of that. Anything that's not mom to baby, we're going to call horizontal. So horizontal is probably the most common type of transmission we have. Okay, so now I don't want you guys to panic on transmission. it's just, I was thinking maybe on the TEAS test or nursing school, you might need to know a lot more about transmission. So this is just for future use, but don't panic. On page 2, I tried to develop a little visual of how your textbook describes transmission. And I'm just going to pull a few key terms from this huge sheet here. So remember, you don't need to know all of this for the exam, just the key points we're going to put up here on the board. So first of all, I just wanted to share with you something that I've always found hilarious. In vet school, we had this old cowboy veterinarian come in to teach us about infectious diseases. And he came in, he had kind of a drawl, and walked with bow legs from riding horses since he was 1 year old. And I remember he said, okay, now remember the 7 F's of infectious diseases. And he goes, flies, fomites, fingers, food, flies. And then he left off an F, and everybody in the class starts kind of chuckling, you know, and I was very naive, so it just bugged me. I just couldn't figure out that last step. But anyway, I figured out fornication. That's what it was. Okay. But anyway, so the 6 or 7 F's of transmission. Now, I don't want you to worry. You can see where the transmission is broken down into contact transmission, where you actually come into physical contact with either a person that's infected or an inanimate object that's infected. Vehicle transmission is when there's some medium like food or water or the air that brings the pathogen to you. And then vector transmission, we studied that in lab, right? So let me just tell you the key points that I want you to know. So there is a term that you'll hear over and over again when we talk about disease transmission, and that is fomite. So I want you to know what a fomite is. So a fomite is an inanimate object, a non-living object that becomes contaminated with the pathogen. And then when a new susceptible host comes into physical contact with that fomite, the pathogen is transferred to the new host. Okay, so fomites can be powerful ways for pathogens to be transferred. And one of the best fomites that we're really worried about are contaminated, reused, what do you think? Yeah, hypodermic syringes and needles. And the problem here is with folks that are using illegal drugs is that they often don't have access to clean, sterile needles. So unfortunately what happens is that people will reuse them themselves or they'll share them. And of course, as you guys know, you probably just got instinct. That's a great way for HIV and the bloodborne pathogens such as HIV, hepatitis B, hepatitis C to be transferred. Contaminating blood with the pathogens remains in the needle and syringe. So the next time somebody else uses it, they end up injecting themselves with those bloodborne pathogens. Of additional concern that we're seeing more and more of in emergency rooms is that when these reused needles and syringes, when they are used, literally, they often are dirty. And what we need to remember is in soil and dirt, we have endospores of tetanus, Clostridium tetani. So what you'll see in an emergency room is an IV drug user presenting with wound, back up you guys, sorry, not tetanus endospores, botulinum endospores. You'll see some IV drug abusers present in emergency rooms with wound botulism. And what happens is the needle gets dirty, it has botulinum endospores on it, so when they end up injecting themselves, they inoculate the botulinum endospores into the tissues. Often they're already infected in abscess, there's not good blood supply, the endospores germinate, start pumping out botulinum toxin, and the person will experience what's called a flaccid or ragdoll paralysis, okay? And they can die from respiratory arrest. So another example of why these reused hypodermic syringes and needles is really good fomites. Okay, so huge concern. Okay, so that's one vocabulary word, fomites. And then, let me see here, was there anything else? Okay, the only other little tidbit, you guys, was on vector, the arthropod vector transmission. You'll see that under the vector transmission, there's two terms there, and one says mechanical and the other says biological. Mechanical vector transmission is when the vector just passively moves a pathogen from one place to a new host. And the classic example is you guys are out in the summertime having a picnic in the countryside, and there's cattle nearby grazing and pooping, and you know wherever there's poop, there's flies, right? So let's say one of those cows is shedding E. coli 0157H7. Okay, in the manure is the bacterium. The flies, the field flies, come and walk all over the manure, and they pick up the manure and the E. coli on their little feet, and then they fly over to your picnic, and they walk all over your potato salad, they walk all over your sandwiches, and they transfer the E. coli to your food, and then you eat the food. Okay? So in that case, that's called mechanical transmission. The pathogen is not growing or developing in the vector, but the vector is serving to bring it to you. The other form of vector transmission there we have listed is biological. Biological vector transmission is what we were studying in lab. For example, plasmodium that undergoes replication development in mosquitoes, that's biological transmission. Lyme disease, which undergoes development in ticks. Yersinia pestis, which replicates inside fleas. So when pathogens require time to grow, to replicate, or to develop inside an arthropod vector, that's called biological vector transmission. And it's most of the examples we used in lab. Okay, all right. So that's all we're going to say about transmission. So remember vertical versus horizontal, remember fomites, and remember the arthropod vector transmission, mechanical versus biological. Okay, so we're going to move on then to step 3, and how to be a good pathogen. And you'll see in step 3, the pathogen has reached a new susceptible host, and now they're going to enter the host. And there's a concept here we want to explore, and that's the concept of infectious dose 50 and lethal dose 50. Now we want to see how those two terms relate to virulence, the severity of disease. And the reference on this one, you guys, is down in the lower left-hand corner. Okay, down here in the lower left-hand corner are the little cryptic notes for this. Okay, so first of all, we want to understand what is infectious dose 50. Infectious dose 50 is the number of pathogens required to infect 50% of a population of organisms. So it's called IV50. And in the second term that I'd like you to know is called lethal dose 50. So what do you think lethal dose 50 is? Good. It's the number of pathogens required to kill half the population of test organisms. Okay. Now, what I want you to have under your belts is that there's an inverse relationship between ID50, LD50s, and virulence. Virulence is the severity of the disease that is caused. Okay? So, as infectious dose 50s and LD50s decrease, what do you think happens to virulence? Virulence increases, yeah. So as we have a decrease in ID50s and LD50s, that means we're going to have an increase in virulence. And like kind of application, what this means is at a really low ID50, LD50, very few pathogens are required to infect you and kill you. You know, like 10, that's a really low ID50 or LD50, right? So that means those very few pathogens, they have incredible ability to harm us, right? So their virulence is really high. Okay, so remember this, you guys. There will be at least one question on this. You understand this inverse relationship on the exam. Okay, so the lower the ID50, the lower the LD50, the more frightened we are of those pathogens, right? It's like, oh no, don't ever want to get those pathogens. Okay, folks, so now what we're going to do is we're going to move actually down to step 5. How these pathogens are going to cause harm to us. Okay, and we're going to focus on bacterial pathogens to start with. Okay, and all we're going to do is introduce bacterial exotoxins. Okay, so what we'll just get started today is a discussion of this table that you know you're going to see some part of this table on exam 3 on a short exercise. So we just want to start working on characteristics of bacterial exotoxins, and then next Tuesday we'll compare them to bacterial endotoxins. So let's take a look at these little toxins here. Okay. So we'll first start with the most lethal of those two toxins, and those are the bacterial exotoxins. Bacterial exotoxins are some of the most poisonous toxic compounds known, meaning with some of it takes tiny amounts to kill us, like botulinum toxin. Tiny amounts can kill us. So we'll just walk our way down this table, just so we understand what the table means. Okay, so when we look at composition means what is the toxin made out of? So by definition, bacterial toxins are all proteins. They're all proteins, okay? And many of the bacterial exotoxins are what are called AB toxins. And AB exotoxins are exotoxins that are made of two subunits. Okay, so there's two subunits. And I'll start with the one that's involved with attachment. So the B subunit is called the binding subunit, and its job is to attach to a specific host cell receptor. Just like we saw with viruses, these exotoxins also must bind to complementary host cell surface receptors. And this knowledge, you guys, is powerful, because that means if we have a bacterial exotoxin and we want to develop a safe vaccine against it, we could use like genetic engineering and just make the B subunit, just make the pure B portion, and use that as a vaccine. You'd inoculate people with it. What kind of antibodies would we describe the antibodies made against the B subunit? Would they be neutralizing antibodies? Okay, and I guess, we need to know, what do we mean by neutralizing antibodies? What do neutralizing antibodies do to protect us? What do they block? Attachment, right? So if we could, we'd have a really safe vaccine if the vaccine only contained the B subunit of the exotoxin. We make antibodies against it, so that if we ever encountered the real exotoxin, we'd already have these neutralizing antibodies. They bind the B subunit. The B subunit can't bind. The other subunit can't get into our cells. So the other subunit, we'll call the active subunit. This is the one that uses the B subunit to get into our cells, and it's going to cause the cellular damage or abnormal function. Okay, so again, for the A subunit to cause damage to our cells or to cause disruption of normal function, it has to get into our cells. And the only way it can get into our cells is if the B subunit binds to our cell first. We block the B subunit from attaching to our cells by those neutralizing antibodies. The A subunit can't get in. We're not going to get hurt at all.
I feel stupid. What is an exotoxin?
Oh, okay. So we're that's what we're describing right now. This term exo means that the toxin is going to be usually, not always, secreted from the bacterial cell. So that's what we're doing right now. Is we're describing what are exotoxins. So they're proteins. Many of them are made of two subunits, A and B subunit, which we just described. Okay. Yeah, I know we're going so fast, you guys. We're just like information overload. Okay. So the next thing there is location. Many bacteria secrete the exotoxins while they're still alive. But some of the exotoxins, like the Shiga toxin of E. coli, accumulate in the cytoplasm until the E. coli dies and releases massive quantities. But usually exotoxins are secreted by living bacteria. Okay, which groups of bacteria can synthesize exotoxins? To make an exotoxin, all you need is the gene for the exotoxin protein. So that means both gram-negative and gram-positive bacteria can make these. So for example, gram-positive bacteria that make exotoxins, tetanus toxin is an exotoxin. Botulinum toxin is an exotoxin. And both of these are made by gram-positive bacteria, members of the genus Clostridium. Examples of exotoxins made by gram-negative bacteria: cholera. Cholera toxin causes a watery, profuse diarrhea. And one that you guys might remember, Exotoxin, that damages blood vessels and can cause bloody diarrhea and hemolytic uremic syndrome. Okay, so we can see both gram-positives and gram-negatives can make exotoxins. Okay. The next line there, you guys, is the toxin heat sensitive. So help me out, you guys. Are proteins sensitive to high temperatures? Will they be denatured? Yes. Yes, they will. So most exotoxins are heat sensitive. That is, they'll be destroyed, denatured by heat. There's some exceptions, but we won't worry about those right now. Toxicity. So toxicity relates to like how much toxin is required to cause harm. Exotoxins, like we said, they're some of the most toxic poisonous substance known, so tiny amounts can cause severe damage. Okay. Effect on body or pharmacology. This means, is it a generalized effect, or are the toxins very specific? And exotoxins tend to be very specific. The tetanus and botulinum toxins act on the nervous system. Cholera toxin acts on the cells of the intestine. Exotoxin acts on cells of blood vessels. Okay, so exotoxins tend to be very specific. This is the last one, you guys. Pyrogenic, that's a vocabulary word, you'll see it on the exam. Pyromaniacs, what do pyromaniacs do? Set fires, right? So what does pyrogenic mean? Fever producing, right? Okay, so exotoxins as a class are not fever producing substances. Okay, and then toxoids are modified toxins that can't cause harm but will trigger a protective immune response. Exotoxins can be made into toxoids. That's part of the vaccines. The tetanus vaccine is a tetanus toxoid. Okay. We inject a modified tetanus toxin. It can't harm us, but it produces neutralizing antibodies. And then those neutralizing antibodies, those protective antibodies, are called antitoxins. So we can produce antitoxins, neutralizing antibodies against exotoxins. Okay? So what we'll do then, you guys, on Tuesday, we'll compare